Hello friends, today we are going to discuss practice school on the domain of pharmaceutical analysis. The course outcomes of practice school of pharmaceutical analysis domain. First, to operate different analytical instruments. Then, to demonstrate calibrations of different analytical instruments and glasswares. To understand the regulatory guidelines. Then understand the responsibilities of quality assurance, quality control, and good laboratory practices. Then prepare analytical records, reports, documents, and results. Then interpret structures based on UV, IR, mass spectroscopy, and NMR data. Next is the course contents, which includes 5 modules. In that, first module 1, that is general introduction related to pharmaceutical analysis, which is of 20 hours. 20 hours allocated for module 1. That is first general introduction of analysis, then different techniques of analysis and the importance, then importance of pharmacopoeia, then introduction to SOP, that is standard operating procedures, and then preparation of reagents and standard solutions. Next is the module 2, that is theory, principle, instrumentation, applications of following analytical instruments, which includes analytical balance, then pH meter, UV visible spectroscopy, IR spectroscopy, fluorimetry, high performance liquid chromatography, then HPTLC, that is high performance thin layer chromatography, then DSC, that is differential scanning colorimeter, then dissolution apparatus, particle size analyzer, NMR, that is nuclear magnetic resonance, then mass spectroscopy, analytical method validation. All these are the analytical instruments. We have to study the theory, its principle, then its instrumentation, and the applications of all the instruments. The next module 3, which is calibration, operation, and handling of analytical instruments. 45 hours allocated for this, which includes analytical balance, calibration, operation, and handling of analytical instruments. That is analytical balance, then pH meter, UV visual spectroscopy, IR spectroscopy, fluorimetry, HPLC, then HPTLC, DSC, then dissolution apparatus, and last particle size analyzer. We have to calibrate all these instruments. We will study how to calibrate the analytical balance, pH meter, UV, IR, and how to operate, how to handle all these instruments, all these sophisticated instruments. 45 hours are allocated for this, only for calibration, operation, and handling. The next module 4, that is preparation of analytical methods for various drugs. 50 hours allocated for module 4, which includes first the selection of drugs. You have to select the drugs. For that, we have to search the new approved drugs on the FDA site. Then collection of monographs, then literature search, that is literature review, literature search, same. Then selection of equipment, then method development. In first, that is selection of drugs. First we select the category, then we select the new drug. According to the drug, we will do the literature search. Then which method we are going to do, that is by using HPLC or UV, that equipment we will select. Then method development. In method development, firstly we have to use the UV. If you are going to develop the analytical method, then first, if you interested in HPLC, there also you have to go for UV spectra. From UV spectra, you will get the wavelength. From that wavelength, you will carry out all the scanning, all the assay. Then next is the method validation. Method validation includes which is we will do the method validation according to the ICH guidelines. There are 9 to 10 parameters like system suitability, then accuracy, then precision, robustness, ruggedness, range, LOD, LOQ. These are the parameters which we have to do in the method validation. And last, the quantification of drugs. Then next module, fifth module, that is report writing. You have to prepare a report on practice school which should not be more than 25 pages. You have to prepare 3 copies of the same. Thank you.